Hi there, our topic is to apply topological methods to visualize and analyze human prefrontal cortex data. Here are the contents that we want to cover today. And as shown in this picture, the golden part is the human prefrontal cortex. We have three motivations in this project. The first one is to discover the complexities of human brain development and the cell differentiation paths. And secondly, we want to identify the subgroups of the PFC cells and trace their developmental trajectories. And thirdly, we also want to evaluate the effectiveness of different topological methods regarding this problem. Particularly, we use PCA, UMAP, T-sign, and the member algorithm. And we also compare their results. And our data is the TPM values. Basically, the TPM say a transcript per million is a value derived from the transcript count of the genes, and essentially it is an indicator of certain the gene's expression level within one cell. And the higher value means that the cell has higher expression on that gene. So this chart shows the distribution of all the TPM values of the original data set. The horizontal axis is the TPM value, while the vertical axis is the value count. And basically, these two axes here are in log level, so we need to normalize the TPM data using this equation. And essentially, the root cause for this is that most single cells have the sequencing depth less than 1 million reads. So we need to normalize first divided by 10n plus 1, and also use a log to normalize the data. So. And to prepare the data for the main cell type clustering, we apply two conditions to filter the original normalized data. So the first one is to exclude those genes with TPM larger than 1 but expressed less than 3 cells, and we also exclude those cells with less than 1,000 genes expressed. Say their TPM most genes have 0 value in the TPM. And to prepare the cell subtype clustering data, we exclude 13 genes which are irrelevant to the subtype cells, and we also exclude cells with high expression level on the hemoglobin genes because they are likely not to be the neuron cells. And thirdly, we select the top 1,000 most expressed genes to reduce our computational cost. And here is the full description of all the data sets involved in our project. Particularly, we label the main cell type based on 6 gene markers, as shown in this table.
We have utilized 3 main methods to conduct the data visualization and dimensionality reduction. They are the principal components analysis, uniform main field approximation and projection, T-distributed stochastic neighbor embedding. And the first is about the principal component analysis, PCA. As we all know, PCA is a linear dimensionality reduction technique that aims to find the directions along which the data varies the most. It achieves this by transforming the original high-dimensional data into a new set of orthogonal variables called principal components. PCA helps in reducing the dimensionality of the data while preserving the most important patterns and variance. And as you can see in these two graphs, the left is about the PCA results grouped by gestational weeks, and the right is about the PCA results grouped by cell types. And it shows that there is no very clear boundaries for this PCA analysis on all data sets. And the results turn out to be better when we utilize the UMAP. The UMAP is a relatively new dimensionality reduction technique that aims to preserve both local and global structure in the data. It is based on the assumption that the data points are uniformly distributed on a main field. UMAP constructs a high-dimensional weighted graph representation of the data, and then optimizes the low-dimensional representation that preserves the topological structure of the original data. So as you can find very easy that you can easily find out that the boundaries in these two graphs for UMAP results are much more clear than the results for PCA. The last method we use is the T-distributed stochastic neighbor embedding. And T-distributed stochastic neighbor embedding is a nonlinear dimensionality reduction technique that is particularly effective in visualizing high-dimensional data in low-dimensional spaces. It constructs a probability distribution over pairs of data points in the high-dimensional space and a similar probability distribution in the low-dimensional space. The technique then minimizes the divergence between these two distributions to create a mapping that preserves the local structure of the data. As you can see in these two graphs, that the T-sign methodology gets the most clear boundary among these 3 methodologies. And as we can know, we observe that the clarity of the boundaries in the 3 methods follows the pattern T-sign larger than UMAP larger than PCA. This observation is consistent with the nonlinearity of these algorithms. Additionally, we notice that the cells of type inter are distributed across almost all the clusters in the graphs, which were grouped by gestational weeks. This distribution aligns with the fact that individuals at every gestational week must possess interneuron cells. However, none of the graphs none of the graph of all these 3 methods exhibits a clear trajectory of revolutions. It's a weakness of these 3 methodologies. And we also utilize the hierarchical clustering algorithm. The prevailing hierarchical clustering consists of 2 main methods. The first is the algorithmic clustering. The second is the deceive clustering. We utilize the first one method to get the 20 subgroups. The algorithm-generative hierarchical clustering method is a bottom-up approach for grouping similar data points together to form clusters. It starts with each data point as a separate cluster and then iteratively merges the most similar clusters until a stopping criterion is met. It provides a flexible and interpretable approach for clustering data by capturing the hierarchical relationships among data points. As you can see in the graph, the branch depicted in red lines represents the 20 subgroups obtained through the hierarchical clustering algorithm. This algorithm clusters the data by capturing the hierarchical relationships among data points. However, it is worth noting that the majority of the captured branches are concentrated in the right portion of the hierarchical trees, which indicates that a lack of diversity in the clustering results. To further investigate the reasons behind this clustering pattern, we need to dive into the detailed data of those branches clustered on the right.
Topological data analysis is a recent branch of data analysis that uses topology and in particular persistent homology. The TDA is used to extract the meaning from the shape of the data. And here we will use mapper, which is the most commonly used TDA approaches to do the data analysis and the visualization. So the whole algorithm can be organized as follows. So we find that there are 4 steps. So first, we need to map it into a lower dimensional space using a filter function. So it can be a projective function or any kinds of the dimensional reduction methods. And secondly, we will construct a cover on the projected space in the form of a set of the overlapping intervals. And in the third step, so each interval, we can cluster the original data. And then finally, we can construct a graph where the cluster sets are connected if they share some points in common. So here we just present the result of the processed data on the mapper. So the output of the mapper on original data is a network where each node is a cluster of cells and each edge means the 2 clusters share some common cells. So on the left hand, we assign color based on different gestational weeks, and on the right hand, it's assigned from the types. So the size of the node is proportional to the number of the cells in the node, and each color of the node represents a specific property of the cells. So by visualizing the shape of the data, we may find the mapper not only separates the cells from the different weeks or types, but also preserves the continuous structure of the single cell RNA sequence data. Although another advantage of the mapper is that you can view data under different resolutions and capture the patterns of the different scales. So which means with the help of the mapper, we can clearly capture the global structure as well as their detailed patterns at a high level of resolution. So after that, we implement out the more specific subtypes clustering and analysis. So all the regional cells could be further divided into 20 clusters by random forest analysis. And by using different color, we may identify their gene expression models with their potential biology meanings, which helps the interpretation of the human prefrontal cortex development. So in the figure, we may find the subtype 12 has many cells in common with subtype 7 and subtype 3. So while the later 2 are located in different branches, which indicates different directions of the cell differentiation. So the same thing happens between the subtype 7, subtype 5, and subtype 8, when we find more details in their nodes. And among all the different weeks, the GW16 has most of the data samples. So we filter them out to observe their distribution over the subtypes. So this figure shows that most of them lay in subtype 12, which comply to the same structure as their whole cell distribution. Additionally, these cells have diverted finite expressions towards the subtype 5 and subtype 9.
So to sum up, we may conclude that the well-known dimensional reduction methods such as PCA, UMVP, they can show the clear boundaries among different clusters, but they cannot retain the topological structure on the original data. But with the help of a mapper, we can preserve the continuous structure for the gene expression and effectively differentiate different cell types. So based on the subtype analysis, we can go further, which means one type of the cell can develop into another type of cell through different differentiation paths, which is consistent with the phenomenon of the cell differentiation. And the whole study provides insights into the human prefrontal cortex cell development, which is very important for the later study.